Welcome everyone to the Lightning Talk session hosted by the Festival of Genomics and Biodata Conference. My name is Joanna Greenhough and I'm a product manager at Promega. Today I'd like to introduce you to Promega's products that support the NGS workflow, including long-read sequencing. The process of next-generation sequencing starts with the extraction and purification of DNA samples. The next step is the amplification of genes of interest in the process of PCR. Target genes from samples are then pulled into a sequencing library, and this can be done using either DNA bar-coded primers or DNA bar-coded adapters for sample tracking. The next step is loading the sequencing instrument, for example, Pacific Biosciences single molecule real-time or SMART sequencer, that is capable of generating reads tens of kilobases in length, which allows the assembly of complete genomes and sequencing of full-length transcripts. As already mentioned, the first step in the next-generation sequencing workflow is the extraction of DNA samples. Promega's Maxwell RSC instrument is a compact, automated nucleic acid purification platform that processes up to 16 samples simultaneously. Using pre-filled cartridges and pre-programmed methods, the Maxwell RSC offers consistent, reliable DNA or RNA extraction in 25 to 60 minutes, depending on sample type. The process is compatible with multiple sample types, such as blood, buccal swabs, cells and tissues, plants and food, fecal samples, FFPE, saliva, and plasma. Our customers refer to Maxwell as a workhorse of their lab that enables high-quality nucleic acid extraction and purification with minimal steps and less hands-on time, and improves the quality of the extracted material. The next step of the process is amplification. The high quality of the DNA extracted by the Maxwell instrument allows for the amplification of even the longest genes that are being studied. This can be achieved using Promega's GoTag long polymerase. Promega's GoTag long PCR master mix contains the high-performance GoTag hot start polymerase in a specially formulated mixture with a thermostable proofreading polymerase. It allows efficient amplification of up to 40 kilobases from lambda DNA or 30 kilobases from human genomic DNA. The presence of a proofreading enzyme to repair DNA mismatches and a highly processive polymerase allows the polymerase to continue to elongate the DNA much further, resulting in longer DNA amplification. The formulation of the GoTag long PCR master mix components enables simple reaction setup, provides consistently accurate and robust amplification of long DNA amplicants, and is ideal for cloning genes, mutational analysis, and DNA sequencing. High-performing polymerase is especially important when amplifying large, variable length or hyperpolymorphic and complex genes. Promega's GoTag long polymerase allows our customers to obtain higher amplification success rate and higher yield with fewer cycles, while the robust hot start feature enables multiple amplification setups without worrying about generating non-specific products. The next stage in the NGS process is the library preparation. Proneg's size-selective purification system can be used multiple times throughout the library preparation process, from cleaning up the initial PCR reaction through to the other enzymatic reactions throughout the library prep. The Promega size-selective purification system enables the rapid and efficient magnetic resin-based purification of double-stranded DNA for next-generation sequencing, polymerase chain reaction, and general molecular biology applications. The Promega system allows users to select the desired size of purified DNA fragments from 100 to 700 base pairs. The formulation provides significantly improved selectivity, reproducibility, and yield relative to traditional DNA purification methods. Also, Pronex beads have significantly lower viscosity than other beads on the market, making them far easier to pipette, increasing reproducibility of size selection and recovery. Pronex beads can be used in both manual and automated high-throughput workflows, and our team of field support scientists is always available to support the automation of Pronex. Our customers have reported that Promega's Pronex beads behave better than other brands of magnetic beads, particularly with some of their PCR applicants where they previously experienced what they described as flaky clumps of beads that had negative impact on sample recovery. When performing these cleanups in a 96-well plate format, having consistent non-slipping beads is absolutely vital. It's also important to maintain continuous optimization of the size selection process and sample cleanup. Another Promega product that can be used in the NGS workflow is the Quantus fluorometer. The Quantus fluorometer is a compact, easy-to-use dual-channel fluorometer designed to provide highly sensitive fluorescent detection when quantifying nucleic acids. The Quantus fluorometer is optimized with pre-programmed settings for Promega QuantiFluor dye systems to quantitate nucleic acids, but also offers the flexibility to create customized methods and quantitation settings for other fluorescent dyes. Quantus fluorometer can be used at a number of stages in the NGS workflow. Our customers quantify the total input DNA into their library prep, but also pre and post the final size selection stage where they need to tailor the reagents to the amount of DNA present and not just the volume. They also perform a sample cleanup step before loading the sequencing machine. Taking into the account any loss of yield from this process allows them to achieve optimal loading of the sequencer, generating high-quality data. As a last step in the NGS workflow, the sequencing instrument, such as Pacific Biosciences Smart Sequencer, would be loaded and the sequencing process would start. After sequencing, various analysis tools can be applied to analyze the long-read sequencing data. As mentioned in this talk, Promega's products not only support short-read NGS workflows, such as those of the Illumina platform, but also the long-read ones like the Pacific Biosciences Smart Sequencing platform. We've also mentioned that one of many challenges for long-read sequencing is that many purified DNA samples contain short DNA fragments that compete for library preparation and may reduce the overall N50 of the samples. The size selection of template can be used to maximize sequencing yield and read length. Conventional bead protocols for DNA cleanup and size selection typically target DNA fragments less than 1,000 base pairs. But with a small change to the protocol, Pronex size selective purification system can be used to eliminate DNA fragments shorter than 4 kilobases without the need for custom buffers or reagents. This step can improve Oxford nanopore long-read sequencing read lengths. This is just a very short summary of the role of Promega's products in supporting the long-read sequencing workflow. If you'd like to discuss technical aspects of our products, and I'm happy to have a chat, please contact me via my email address, joanna.greenhough@promega.com. Thank you.